So the next session is going to be on infectious diseases, and the first speaker, who is also a co-chair for this session, Professor Yukinori Okada, who is from Riken Center for Integrative Medicine. So he's going to talk about statistical genetics, elucidate biology, infectious diseases.
Okay, hello everyone. This is Yukinori Okada from the Riken Institute in Japan and also University of Tokyo and Osaka University. So this session is for infectious disease. Actually, my talk is, as you can see, more genetic side. So I'm going to tell you how genetics think of these single cell stuff. So anyway, for us, human genome and the epigenome is the blueprint of humans. So if you can fully understand the full meaning of your sequence of yourself, you can reconstruct yourself. But we only know, we can easily sequence, but we do not understand what is happening. So our study is how we can decipher the blueprint from, and how we can extract biology from the tip to the bottom. And our field, we are always evolving the new technology. So technology always more rapidly develops than we expect in both and dry vessels. Here's the history of the last 15 years, how associations study DNA and RNA stuff. Everything changed quickly, but I know 15 years later, students feel left the single cell sequence technology today. So especially, anyway, implementing this technology is important. And this is really easy to say how to do this, but keep trying. So anyway, new technology is important. So I'm going to tell you today the 3 short stories. So how is the large-scale human genetics things? So what is happening in human genetics is just super big. The Human Genome Project became bigger and bigger. Especially UK Biobank has released 500,000 individual genome and phenotype data freely. So this changed how to do the games. And especially we have tons of GBUS and tons of association studies of hundreds of phenotypes. And I have been working on more biobank stuff for GBUS stuff, especially in Biobank Japan, which I'm now working as a core member. So this is one of the latest catalog of trans-biobank GBUS meta-analysis from Biobank Japan, the UK Biobank from UK, and FinGen from Finland, and also collaboration with Tohoku Medical Megawatt. We have conducted GBUS more than 200 of phenotypes, and when we have so many GBUS summary statistics, we can extract biology by cross-populational way. But anyway, what is most important is GBUS summary statistics should be publicly available without any restriction. So we have constructed database. You can freely get any kind of summary statistics from these databases. But we geneticists have been really interested in the cell type specificity stuff. So this is the figure 5 years before we construct. So integrating phenotype GBUS summary statistics and cell type specific epigenetic motifs, we can finally deconvolute the connection between human complex strain cell types networks. You can see that the regulatory T cell related to Graves' disease or asthma is related to the Th1 cell. It makes sense. The point is we are not using any prior biological or immunological knowledge. Just using genetics and human epigenetics, we can now start to reconstruct biology. So this is an interesting field. And as I said, technology always develops, so getting more new omics with single-cell resolution data, this figure getting more finer resolution. For example, the immune cell stuff, we have recently updated their network. So this is the immunophenotype result with 46 immune phenotypes of the peripheral blood and 11 autoimmune diseases. Anyway, we can get how they contribute to each other. For example, even within regulatory T cells, memory T cell is increasing in systemic lupus erythematosus, but naive T cell is increasing in psoriasis. So anyway, once you get more detailed figure of human cell atlases, we can project them into human genetics and get more biology. So this is how to project human cell atlas to the human genetics. But recently, we have another type of approach, project GBUS into single-cell stuff. For example, this is the work by Gosato from our laboratory. We just conducted a pan-cancer GBUS in Biobank Japan, UK Biobank, and found that shared genetic backgrounds between prostate and breast cancer. So these are two cancers do not occur together because of the barrier of the gender, but they have shared genetic backgrounds. But when we project these genetics into single-cell resolution, we get some shared genetic and serotype specificity, especially in epithelial cells. So now we are starting to project human genetics into single-cell size. Anyway, what is most important to make the data publicly available? So field.jp is our interactive website. So if you can put your phenotype name or gene or variant name of interest, because this is infectious disease session, just let's put pulmonary tuberculosis, you can get Manhattan plots of it. And you can see the strong hits in HR genes in MHC region. Yes, this region is a critically important region where we want to understand genetics, genomics, and single-cell omics of infectious diseases. Let me talk a little bit about this region. So this is the MHC region. So, but generally, fine mapping causal variants within the gene-associated oocyte is important to understand mechanisms from variant to function in disease biology. We have several talks in this session today. But especially HA gene in the MHC region, highly polymorphic reverse genotype. So we have conducted in silico HA variant imputation studies in Japanese population and conducted fevers and found they have strong impacts on much more wider range of phenotypes, including many kinds of infectious diseases. And yes, we have recently updated these references. So we resequenced HR gene and KR genes of in Japanese population and construct in silico pipeline to highly genotype them and machine learning analysis classify them. But anyway, these HR and KR imputation reference model is publicly available, so you can freely use it to impute your data of the variant. This is critically important, especially even in the single-cell eQTL stuff. And also, we have recently updated how to impute HRE genes. So previously, we used MC Markov models, but we have implemented the deep learning, especially convolutional deep learning method, just converting population genomes into images. So this deep learning-based HRE imputation named deep HRE was really high performance, especially when accurate imputation stuff rare HRE variants. So how to apply convolutional deep learning to the human genetics is one of the topics. Actually, in this task, the deep learning was working very well. So anyway, human genetics is thoroughly integrated in the infectious disease genomics. So I'm next going to talk about our infectious disease example in COVID-19 pandemic. So you may have sometimes seen this Manhattan Pro, famous one for COVID-19 GVAS in Europeans. Yes, here's a strong signal in chromosome 3. So this is the power of GVAS, the hypothesis-free approaches. Even in the situation we didn't know what is COVID-19, we can get the pieces of the evidence which is related to COVID-19 biology. So the point is, yes, this variant is highly polymorphic, but not polymorphic in East Asia. So we definitely need to make our own resources to understand what is COVID-19 in East Asians. So we launched Japan COVID-19 Task Force, which finally consisted of more than 100 hospitals all over Japan. Actually, the Dr. Namgun Fu, who joined the session yesterday, led this project. And we finally collect DNA, RNA, cell from more than 6,000 affected individuals. And we are now conducting massively the much more the omics data and omics analysis. For example, one of the initial report from the GBAS focusing on younger and severe patients, we found one signal at chromosome 5 at DOX2 gene locus. So DOX2 gene has an immune cell-specific expression profile, which regulates LAC activator, regulates lymphocyte activation, macrophage mitigation, type of interferon production. So it means that, yes, DOX2 was one of the center of the virus responses, and deficiency is mainly disorder. So we try to show 2 points. So we need to show DOX2 is surely depressed, the function is low in the severe COVID-19 patient. And secondly, when we perturb the function of DOX2, if the pneumonia gets worse, and based on these two findings, DOX2 should be the causal gene for the severe COVID-19. So we then try to apply, because we have a new disease, we try to apply new technology. So we used the single cell technology. Actually, it was almost the first time for our laboratory to handle the single cell omics. But yes, this is a peripheral blood single cell result. You can see DOX2 is highly expressed in non-classical monocytes, CD16 high monocytes. Yes, that's the center of the interferon responses. And you can see they are decreased in non-classical monocytes in severe patients. And if the patient has the risk area, they get the decreased dosage, especially under the COVID-19 infection state. So the cell type specific and a context-specific equity effect of the variant in toxin expression. So single cell, and it clearly shows the variant of function mechanism in COVID-19 severity. And yes, we then try to inhibit toxin using CPIPP inhibitor, and clearly shows that when we put CPIPP, the serum hamster infection will get worse and die, and weight loss, lung edema, enhanced virals, impaired macrophage recruitment, and dysphilic type interference. So doctor was survey the responsive regime of COVID-19 severity and told what was happening in the pneumonia. So omics analysis always tell us some piece of the biology of COVID-19. So we next constructed the bulk RNA sequences. So we conducted eQTL analysis of COVID-19 patient. Especially we focused on interaction eQTL, iEQTL is how the eQTL effects changes based on COVID-19. This is because bulk RNA sequence achieves, we can get sample size, we can have enough power to identify iEQTL. Especially the COVID-19 iEQT loss has mostly specific to neutrophil EQT result losses. It means that neutrophil activation was showing increase in the COVID-19 activation. And the next omics strategy for us, the proteomics. We then conducted all-link 3,000 protein omics assays in Japanese population, more than 1,000. So we conduct much more EQT and PQT analysis in the same individual COVID-19 Japanese. And actually we found that, yes, EQT and PQT is similar but different. And actually considering both is important to confine the drug targets. And we also conduct the NPRA assay to confine the genetic parts. But I think single cell EQT is fine, but single cell PQT may also be the important topics next. But when we want to try to confine cell type specificity, single cell RNA sequence is the best technology today. So yesterday, the hero from my laboratory talks, I don't talk the details of it, but single cell sequence analysis in Japanese population finally found that the monocyte and dendritic cells, the innate immune cells are the causal roles of the COVID-19 patients. So applying such existing method is fine, but we then try to find new technology for single cell analysis. So Yoshihiko Tomofuji from our laboratory found the new method SC-Linux. So it's X chromosome. One of the X chromosome in humans activates, which we learn in textbooks. But up to 20% of gene X chromosomes escapes from HCA. This HCA escape contributes to the differential gene expression between sexes and also differential dosage of the GBS risk variant X chromosome. But it's still difficult as how to quantify escape. So we conduct SC-Linux, which direct observation is for HCA in physiological condition, almost single cell resolution. It's also applicable to export from data. So here's the pipeline. So it's complicated, but very beautiful figure. So nice student always make us the nice method. But anyway, we can now quantify HCA escape from pipeline single cell and also multi-omics data. Then we are now applying this method in collaboration with as an immune diversity address IDA network and there's open data. And we found that here clearly shows the differential expression, gender-specific differential expression profile between the X chromosome. And actually we found that HCA escape can explain such different sex differential expression profiles. And then we then checked how they difference across the population. So we checked tissues. We used human tablet sapiens database and found that HCA escapes mostly focusing on lymph nodes, salivary gland, or spleen. It means that lymphocytes or organ which includes more lymphocytes is under the strong HCA escapes, and they also linked to the GBAS dosage effects between sexes. So in the field of the world of single cell sequence, we can extract so many things from even existing data. So it's a really interesting, beautiful world. Okay, let's talk finally from the microbiome. Because we have been talking genetics and genomics, but microbiome of pathogen site is also important. And here comes a new technology. So 16 RNA ribosomal we have been doing, but now we're changing to the metagenome shotgun sequencing stuff. And we are now applying to how to construct the in silico pipeline to utilize shotgun sequence data. So this is one of the initial metagenome-wide association study comparing cases and controls of microbiome. And so we see that maybe, yes, so it shows the bacteria, the size represents dosage and color represents case control. And the most final scale, L7, species level, you can see that some species related promoters increased in the gut of the rheumatoid arthritis patients. So we learned that RA is the disease of joint inflammation, but what is happening in your gut is also associated with the inflammation status. That's very interesting. So we then starting to do many shotgun sequences of Japanese, but found that we still lack the catalog of microbiome even within Japanese populations. So Yoshihiko reconstructed the Japanese gut metagenome data from our own databases, and actually he constructed 20,000 bacteria and 30,000 viruses. Around half of the viruses new findings, and actually we made a catalog Japanese microbiome within a virome and metagenome, and which provides so many population-specific features. For example, what we eat is defining our gut. So we found Bacillus subtilis. This is derived from natto. Natto is fermented soybeans. And we found many bacteria which have beta polyphyllase. It's a nori liver. So what we eat clearly defines our gut, but I think it still changes because of the eating Indian curry the last several days for me. But anyway, this kind of microbiome stuff should be diverse with different across even within agents. So we should definitely change the make the catalog of this data. And the other layer is viromes. So we have conducted a pipeline to quantify the phages, so especially class-like phages and their subfamilies. We found that specific subclass of phages, class-like phages like alpha type, strongly different across disease status. So they are stronger decreasing the RA or SLE autoimmune disease, but increase case corrector cancers. So we still do not have the catalog of these phages in gut, but we should definitely check these status, and they have strong meaning bacterial the virus network within our gut. So anyway, we need to check the network between diversity of human genome and diversity of the metabolome and diversity of microbiome within single population. So we recently reported a database which connecting the diversity within these three modalities. So actually these three are connecting. So genetics and metagenome is not so strongly, microbiome is not so strongly connected, but some of the genetic variants affect your microbiome. So the current our project is how to connect these diversity in such omics data with the diversity in the single cell data. So how they are affecting each other is of interest. I want to show some small scale example. ABO gene variants. It is ABO blood type is strongly affecting your gut microbiome, and ABO blood type is diverse with populations. So there exists some genetic variant which affects your microbiome stuff. And along with such efforts, we found that such human DNA fecal samples include shallow dosage of human DNA. We made a construct to re-identify gender and ethnicity and individual based on this shallow DNA in the fecal sample. So especially human genome sequencer should be properly created as a personal information when uploaded to public database. I think this is also true for the single cell data because we can identify individual based on the human genome variants on the single cell stuff. Okay, finally, but one of the most important tasks for the community is not only research but development of young scientists. So in every summer, I made a 3 days course of the surgical genetics to teach the students or sometimes doctors how to do Linux or how to do software GBA sQTL. But I think it is truly important to make the community wide and also start such biomass course in the medical department of Osaka University Tokyo. But when we do this time, what is most important is we need public data. Public data with fast individual genome data we can use without any restrictions. So without this data, it is difficult to have a high level of education for the community. I think this is one of the definite important tasks for the HCA community to prepare such data for training to wider range of the young scientists. Okay, finally, I would like to say thank you to my lab members and also all the IDA members and for Namgun for the leading COVID-19 research. Thank you for listening.
So, is there any question? So, one or a few questions from the audience? So, I have one. So, do we know the underlying mechanism of X inactivation escape? Is it, just cell intrinsic, like certain cell would have those, or is it due to like stimuli from the environment? So that's why it's in the immune cells.
It's still under discussion. We didn't know figure, but we just starting to able to quantify such phenomenon. The next step is how they are interacting with other environmental, other such outer factors, which is still on the discussion point. Yeah.
All right. Thank you so much. So, so the next speaker is Dr. Veena Patil. She's a principal investigator at National Institute of Immunology, and today she's going to talk about single-cell multi-omics analysis of viral antigen-specific memory T cells in humans. Welcome.
Okay. Would like to start by thanking the organizers and Dr. Patha for this great opportunity to present our work here. So I've been here at NII for close to 5 years now, and I'll just briefly introduce you to my lab and talk about antigen-specific, viral antigen-specific T cells, how single cell helped us in understanding their characteristics. Okay. So this is largely what my lab does. The lab is distributed into 2 different arms. One where we are trying to understand the basics of T cell memory development to various infections, where we try and understand what are the types of memory, what's the developmental lineage, the specificity to different pathogens, and the heterogeneity. And we largely use various omics tools to bring out the gene expression as well as the regulation. So in the other arm of the lab, we are trying to understand the sepsis in neonates by looking at a longitudinal cohort to see if we can predict a prognostic or a diagnostic marker about the babies, which babies go on to develop sepsis, and the ones that develop sepsis, which go on to have a better outcome versus an adverse outcome, and compare them with that of the healthy age-matched neonates. And this is an ongoing project currently in the lab, so maybe we'll talk about it next time. So for today, I'm going to focus mostly on the T cell memory part. So all of you know that whenever there is a disease, it is going to develop an immune memory that leads to the natural immunity. And this is what exactly has been exploited for the vaccination, where through vaccination, we induce immune memory and eventually the natural immunity, and that eventually controls the active disease. Okay. So for people who are not really T cell biologists, I just want to introduce you a little bit to what T cells. T cells recognize the pathogens through their T cell receptors, which are present, the antigens that are presented by the antigen presenting cells through MHC molecules. Upon this recognition, there is a second signal that comes up where there is the co-stimulatory molecules get activated, and the third signal comes from that of the cytokines. The T cells have a dichotomy. There are majorly two types. One is the killer cell, another is the helper T cell. And helper T cells provide help to various other immune cells, including that of the CD8 cells, which get activated and make these cytolytic molecules and kill. I don't have to introduce this aspect because you already heard from multiple speakers today and yesterday that there are cytotoxic CD4 cells, and these are relatively less well studied, whereas the CD4 cells are better understood for their helper function as classified based on their cytokine melee and the transcription factors they express. So the T cell response is a multi-step process where the T cells rapidly expand, which coincides with that of the increase in the viral load or any pathogen. And upon the resolution of the infections, the T cell contract and leaving behind a very small portion of the cells as memory. and that's exactly what we are going to talk about today. So while some individuals are maintaining this memory better, the other individuals fail to do so, and they actually form the protective and the susceptible immune response respectively. So upon a secondary infection, if you have maintained the better immune memory, you're going to mount a better immune response and control the infection subsequently. So now if we want to say there is what is a good memory, so the memory has to be long lasting. Once you have an infection, it should really be there for very, very, very long time. And it should have a potential to differentiate into effector cells so that it can control the infection whenever there is a secondary infection. And should be able to move to the site of infection, that's the mobility, and be able to self-renew even in the absence of antigen. Okay? So in CD4 cells, while the other classification of TA helper cell, which is Th1, 2, and 17, has been better understood, the classification based on the developmental stages of the memory has not been that well understood. But out of that, there are markers which distinguish them into different developmental stages, like stem cell-like memory, central memory, effector memory, and then effector memory expressing CD45RA. We and others have shown that this TEMRA compartment is indeed enriched for the cytolytic molecules and forms a bona fide CD4-CTL compartment. So, sorry. Yeah, so in my previous part where we did using single cell, we showed that though this was thought to be terminally differentiated, it actually was heterogeneous, and we found a long-term kind of memory within this TEMRA compartment. And further, using the T-cell receptor sequencing, we have shown that this actually is the precursor to that of the CD4-CTLs. So if you visualize memory, what is the developmental lineage? Is it a very linear developmental stages, or at every stage there is a bifurcation that leads into a different kind of memory? And what are the specificities to different infections? But in reality, what exactly exists is this kind where you have heterogeneity within each of these compartments. And this heterogeneity could be as subgroups within the memory that can indicate a developmental lineage, or it could also come from the specificity. So for specificity, we have to look into antigen-specific cells, and the characterization of antigen-specific memory T cells is relatively difficult because of their rarity. Okay? So you imagine that you are going to get 10 to 100 cells out of 1 million PBMCs. So it's a very, very rare population. So for that, what we did to be able to understand the specificity, we took the PBMCs from the donors who were seropositive for various infections like CMV, dengue, and then or influenza or COVID, and then also tested them for their responsive to this, the peptide pool stimulation, and sorted the activated T cells based on the surface expression of co-stimulatory molecules such as CD137 and 154, and sorted these activated cells across different CD4 memory compartments and pulled them in a ratio so that we have a representation from each of these memory compartments. To be able to compare the CD4 CTLs to that of CD8 CTLs in the same environment, we also sorted cells from the CD8 compartment, which was the effector memory. Then we performed single-cell RNA-seq, and we multiplexed using the H2Os for each donor, and we also added some of the memory markers as the oligotagged antibodies to be able to distinguish them in the single-cell data, and then we sequenced them. So for now, I'm going to mostly focus on talking about the CMV, and at the end, I'll show how this is in relative to the other viral infections, what is the characteristics. The CMV, we found about 9 clusters, and they were distributed across the different memory compartments. We had, the effector memory and as well as the long-term memory, and they were also aligned with the antibodies that we used, oligotagged antibodies that we used in the, while doing the experiment. So this suggests that we had a representation from different memory compartments. And then we looked into their functional characteristics in an unbiased way. Sorry, it's actually moving very fast. We had several clusters which belonged to which had a signatures of long-term memory, and also we had a cluster which had the cycling cells, a very small portion of the cells as cycling cells. And we had, interestingly, a cluster which had the interferon response signature and a cluster that was showing T regulatory signatures and the cytotoxic, that is the CTL clusters. So, very interestingly, the Treg cluster showed the expression or coincided with that of the interferon response signature. So if we look further into that cluster, so you see that the co-expression of the Treg specific markers show that this is indeed a Treg cluster. And they actually had an entire signature of Tregs enriched in those clusters, and that had an overlap with the interferon response signature. So the next question is, are these Tregs suppressive in nature? And many of the molecules that are linked to their suppressive function were actually enriched in this cluster. But however, they did not really make suppressive cytokines such as TGF-beta and IL-10. We have many other evidences. In the interest of time, I'm going to skip that. We also saw that these clusters express GATA3, not the TBET21 or the ROR gamma C, indicating that they're potentially Th2 suppressive Tregs. And if anyone wants to hear more about it, I'll be happy to talk later. So the next one we focused was the cytotoxic clusters, where we had 2 cytotoxic clusters. The question is, what is the difference between these 2 cytotoxic clusters? And that actually indeed coincided, the CTL signatures coincided with that of the TEMRA signatures. And you can see that the cytotoxic clusters expressed granzyme, porphyrin, which are known for their CTL function. And many of the molecules were commonly expressed, for example, porphyrin and NKG7, whereas these 2 clusters show variability between many of the transcripts that we observed. One was granzyme. The granzyme expressing cluster also made the cytokines, while the other cluster expressed a higher amount of granulysin compared to that of granzyme, and they did not really make the cytokines. And the late effector specific transcription factors like ZEB2 was expressed in the cluster that was making cytokines. The other cluster was expressing a slightly earlier transcription factor like EOMS. So indicating that they're probably at a different developmental stages. We call them pre-effectors and effectors. Sorry. Yeah. And not just these, sorry, I don't know why this is. Yeah, not just this, the interferon gamma and TNF cytokines, they also made many chemokines which were highly expressed in case of effector cluster compared to that of the pre-effector cluster. Next, to be able to say that these are effector and pre-effector, we looked at the T-cell receptor repertoire. Both of these clusters were clonally expanded. And you can see that some of the clones were found in as high as 144 cells. So it's a very highly clonally expanded population. Now, if I take only the top 25 most expanded clonotypes, we found that this proportion of the cells expressing these top 25 clonotypes were more in cluster 2, which is the effector cluster, compared to that of the pre-effector cluster. So the next evidence we wanted to ask was, are they developmentally linked? So we did a pseudo-time analysis with the clusters that are somehow associated with the CTL cluster. One was we used the cytotoxicity signature to take those cells, and the sharing, clonotype sharing between 2 different clusters that shared clonotypes with that of effectors, that the cytotoxic clusters with the any other cluster. And based on that, we used cluster 0, cluster 3, cluster 7, and 2 and 4, which were the cytotoxic cluster. As you can see that the effector and pre-effector clusters really bifurcate, so indicating that they're probably at 2 different developmental stages. Okay. Yeah, so the next question is this difference that we are seeing in the pre-effector and effector because of the CD4 and the CD8 proportion? And both these 2 clusters expressed both of CD4 and CD8 transcripts, indicating that they're actually clustering together. Now, is the proportion of the 4 and 8 different? That is why we are seeing this variability. So we looked at the proportion of CD4 and CD8, and those who are thinking that there are a lot of double negative cells, these are sorted CD4 and CD8 cells, we could not assign them to 4 or 8 based on the transcript expression of CD4 or CD8. So we are calling them as NA, not assigned. And we saw that the proportion did not really drastically differ between these two clusters. And we compared these cluster 2 and cluster 4, that is the effector and pre-effector cluster, and divided these cluster based on the CD4 origin and CD8 origin. And the transcriptome profile within clusters 2 for 4 and 8 was similar, the same way for the cluster 4, suggesting that the difference that we observed was not really because of 4 or 8 proportion, but it is the different stages of the development or their effector molecules that dictated their two clusters. Okay. To summarize, from CMV antigen-specific cell, we found that the cells were at different developmental stages. A lot of people use for the antigen isolation cytokine as a marker. Maybe that's not the right way of looking at it. And the suppressive Tregs were suppressive in function. They also had signatures of interferon response. And the CTLs could be classified into pre-effector and effectors based on their effector molecules that they expressed. And these different types of CTL clusters were not really based on, the distinction was not based on the CD4 or CD8 proportion. So the next question is this a characteristic feature of CMV alone, or it is also found in the various other viral infections? So we looked at 2 other viral infections, that is dengue and then flu. And in dengue, again, we found the clusters between late effectors as well as the early long-term memory. And the signatures again showed that there are multiple cytotoxic clusters which could be distinguished based on their cytokine secretion or the molecules that are linked to their longevity. And we also had a Tregs which were enriched for the interferon response signature. And the CTL clusters were not distinguished based on the CD4 or CD8. The same is true for an influenza virus, where we looked at there were again multiple cytotoxic clusters, and the Tregs were enriched for the interferon response. The CD4 and CD8 did not distinguish these CTL clusters. So overall, the antigen-specific viral antigen-specific Tregs are indeed suppressive in nature, and they are showing an enrichment for interferon response. And we have a lot of evidence suggesting that they could be Th2 suppressive in nature. And the CTLs were clonally expanded and came in multiple flavors. And this flavor could only be distinguished based on their effector molecules and not based on their CD4 or CD8 origin. With this, I would like to thank people who have been very instrumental in completion of this project. The project was led by my first graduate student in the lab, Ronakkar, with the help of other students in the lab, Kirti, Shreya, and Somdev. We have collaborator Anish, who helped us initially to set up all the pipelines. And Dr. Anjali Sharma is from where we get our clinical samples. And the volunteer blood donors, without who's volunteering to donate, we wouldn't have been able to do any of this. And the funding from Welcome DBT India Alliance has been of a great help, where it's a very flexible funding. They allowed us to do whatever we wanted to do. And the sepsis project is funded by Department of Biotechnology, Government of India, and NII for both financial as well as the facility support. With this, I would like to thank all of you for listening, and we'll be happy to take any questions.
We have a few minutes for a couple of questions from the audience.
Great talk. I was wondering, as a virus changes its forms and the proteins change with time, like we saw in COVID and in influenza as well, do you think there might be a change in these cell types and the way they sort of transform themselves from early to late memory, and how to capture that?
Maybe looking at a longitudinal cohort from different stages of the disease may actually help address, but this data set is largely from a blood bank donors, where we actually check their IgG and IgM to make sure that they don't have an active infection. We only took donors who were IgG positive. And also for flu, we saw that they were not IgG positive, but they were peptide responsive. So we are using a pool of peptides, so it may be very hard to say whether there is a viral evolution that could have caused any kind of variation. Then I think we may have to look into specific peptides of the whichever changed and to understand that kind of.
Thank you.
So you tested different peptides or viral peptides. Do you see any peptide-specific TCR responses?
Right.
And have you looked at potential linkage?
So we haven't looked at individual peptides. We stimulate them with pool of peptides, and to again not restrict ourselves to and the cytokines, we use the co-stimulatory molecules to look at it. At this stage, we really cannot say whether this is the TCR we have for this peptide. We tried to use pre-existing some where they have put multiple peptides of different viruses to see if we can predict these TCRs to which peptide this must be responsive to. We didn't really have a lot of success with that because that had a lot of COVID peptides. Didn't have much of the virus that we are looking at.
Yeah, I think now there are tools where you can put barcoded peptides and TCR linkage. That would be very interesting.
Yes, I think the tetramer, but then I think we will again restrict ourselves to a couple of or 2-3 more predominant peptides. Here we didn't want to restrict ourselves to that. That's why we used pool of peptides.
But I think it's even more interesting if you can pool different peptides with different barcodes. So
Yes,
you can even
yes,
do
yes.
this in a very single experiment, I think.
Yes, yes. Yeah.
Thank you.
Yeah, thank you.
Thank you for the nice talk, Dr. Veena. I have a question that I was wondering if we see the T cell type distribution and try to correlate with the memory. As you said that memory should be long-standing. So I was wondering that for the different span of memory, to expect different kind of T cell distribution?
So long-term memory has always should be very limited. So these are all, the effector memory is, if you imagine it should be something that is ready to respond to. So we would, but you don't want to have a lot of effector cells because that's not really positive for the body. So we want to have very few cells, but they should be able to rapidly multiply into an effector kind of cell. So at this stage, especially for the CD4 cell, the temra is a very small compartment, whereas CD8 cell temra is the major compartment. So it has to be, depending on what kind of infection you're looking at, it will depend on that as well.
Yeah, I think that would be like
sorry, we are a little bit behind the schedule, so maybe keep discussion after offline. Thank you.
Thank you.
Okay, thank you for your great talk. Sorry. Okay, the last two speakers of this session are Yukinori Okada going to have the chair. The next speaker is Professor Hideki Ueno from the Kyoto University, Japan. He's going to talk on human liver origin, immune cells, and the ultimate bioage. So he's a really famous immunology professor in Japan. So we are really happy to have you in here. Please go ahead with your presentation.
Thank you very much. I feel honored to be here and to talk about our studies. So I'm Hideki Ueno from Kyoto University. I'm a human immunologist. So basically today what I want to do is I want to basically show what we're doing in my lab in Kyoto University that just established just 2 years ago. We started from scratch. So just really I just want to show the ongoing efforts to achieve to understand what is really a human immune cell in a healthy human livers and the diseased liver as well. So immune system needs to be tightly regulated because the immune system basically is there to eradicate different kinds of microbes. But excessive immune response causes diseases like autoimmune disease and allergy. But the immune system is insufficient, and that also causes diseases like chronic viral infections or primary immunodeficiencies or vaccine failure. Also, immune system can be altered in kind of different way in different diseases like aging, cancer, and neurodegenerative diseases, and cardiovascular diseases. So in that way, so the immune system needs to be really tightly regulated for the full functions. That is also the same in one tissue, in this example, is the liver. So that in the liver, if the immune system is excessive, that cause like different kinds of autoimmune diseases, such as autoimmune hepatitis, and primary biliary cholangitis, and primary sclerosing cholangitis. But also, if the immune tolerance is too much, that again, that causes a problem in the liver diseases, like a chronic hepatitis B infections, hepatocellular carcinoma, or hepatoblastoma, like liver cancers. So to understand the pathogenesis of the liver tissues, so we need to understand the liver resident immune cells, because immune cells is so much involved in many different types of diseases. So what kind of human liver samples can be used for research? So one is a liver needle biopsies. So many surgeons are basically putting a needle and they get a piece of the liver, and that can be do a pathological assessment, and also we can do some single cell sequence analysis. And also some doctors are doing liver fine needle aspirates. Basically, they put the needle under the guidance of the echography, and they target one fine part of the liver, and they suck a little bit medium. And that contains some cells and tissues. And so here what I want to talk about is the liver perfusate. It's, that can be taken during liver transplantations. So in Japan, there's many cases of living donor liver transplantations for like end stage of the liver disease patients. So when you take a donor liver, and before they transplant it to the recipients, this liver needs to be washed out, the immune cells. This is called perfusions. So also we are doing like excised disease liver taken out from recipient patients, and we are taking this liver and doing these perfusions. So here, let me explain what perfusion means. So here's a portal vein of the liver, and we put a tube here, and liquid, basically we put 2-liter liquids into the liver so that we can wash out all the immune cells resident in the liver tissues. So here I'm showing the one example of the hepatoblastoma after perfusions. As you see that really the liver is washed out, they can look like a white, and these black dots, they are all hepatoblastoma tissues. So this really indicates how efficient it is to wash out all the kind of immune cells in the liver by perfusions. So we are getting liver perfusate from this healthy donor liver perfusate, as well as a diseased liver perfusate. So what kind of cells we can collect in the liver perfusate? So here I'm showing the scheme that maybe you saw several times in this symposia. So we are putting liquid from portal vein and washing out a cell from a central vein. So here what is between these sinusoids, and we can wash out all the cells. Actually, this is a place in the liver where that many immune cells are resident. So including T cells, B cells, and the Kupffer cells, it's like a macrophages. So it's a really efficient way to collect a large number of immune cells from a human liver tissues. And so we started this for 2021, as I said, we start from scratch, and we collaborated with the surgeons of the transplantation department at Kyoto University. And so now we have collected more than 80 cases of a healthy donor liver perfusate, the age from 25 until 65 years old. So they act as a donor for the two babies, to survival of the family members. And also we collected liver tissue from the same donors' liver tissues. So that's the paired liver tissues, always the liver perfusate. And also, as I explained, we are collecting excited liver disease perfusate. Now we have collected more than 50 cases. And the disease is really variable and containing PBC, PSC, AIH. This is autoimmune liver diseases, and plus NASH, end-stage NASH patients, and plus HCC patients. And also because we are getting whole liver tissues, so we have a bunch of the liver samples as well. And also we are collecting some needle biopsy samples in collaboration with the Kobe University gastroenterology doctors. So what do we do after we get these tissue samples? So basically we use a multiple platform single cell analysis approach to really analyze the immune cells in the liver. So one thing is, as an immunologist, that's really immunologist's favorite, it's a flow cytometry. And we have a Cytek Aurora and a Sony ID7000 that permits us to play with more than 30 color multiple complex panel for the flow cytometry. And second, we do a single cell sequencing by using liver perfusate and liver tissue, the immune cells. And also we do a single-cell spatial gene analysis. In this case, we use cosmics from NanoShrink. So we integrate basically these three platform analysis to fully understand the biology of the immune cells in the human liver. So there's a kind of advantage, disadvantage for these three approaches. So our favorite ultra multi-cap flow cytometry, the strength is a relatively low cost as compared to other assays. And we don't need so much time to analyze the data, like 1 or 2 days is enough. And that can be really high throughput platform. And the weakness is limited parameters. So if we miss one antibodies to figure out the markers, so we cannot really go back. That's the limitations. And we do many single-cell gene analysis. The strength is really comprehensive, and we can combine that single-cell T-cell sequencing. The weakness is still relatively high cost, and still it takes more than weeks to get the data analyzed. And also we do a single-cell spatial gene analysis. Of course, the strength is of this assay, the spatial analysis, that really locate the immune cells in the tissues one by one. And the weakness is a relatively high cost. So let me go through one by one. So our experience on this flow, three different kind of approaches. So here I'm showing the result of the flow cytometry of the human liver perfusate from healthy volunteers. So as you see, we have like 30 multiple colors in this panel, and we can really demonstrate the complexity of the immune cells included in the human liver perfusate. And to our surprise, this is a frozen thawed perfusate, and we're surprised to see that many kind of neutrophils still present after freezing thawing, so that can be used for deep analysis. And also our surprise was Kuppa cells. Their macrophages, they tend to die after freezing thawing, but really they can survive after thawing. And that will also contain many DC subsets, and the T cell subsets, and NK cells. And the advantage of the flow cytometry is it's really easy to do, and we can compare like a normal liver versus cirrhotic livers. So here I'm showing the example of the comparison of the subsets immune cells. So what we found here is that the cirrhotic liver contains much more like a monocyte macrophages like Kuppa cells, and also that contains some granule cells and the basophils and these subsets. But basically they have less lymphocytes like T cells and the B cells as compared to normal liver cells. That's a relative increase of the APCs, antigen-specific cells. And also to our surprise, the flow cytometry analysis permits us to identify the presence of the hepatic stellate cells, which is supposed to be outside the sinusoid cavity. But still somehow, by using the liver perfusate, we found we can analyze the hepatic stellate cells, both activated and quiescent cells in the FACS. So that's really, that's telling us liver perfusate really fundamental resource to analyze human liver immune cells. And also here we did single-cell RNA sequencing to compare healthy liver perfusate versus cirrhotic liver perfusate. So here we did 3 donors combined, and we did integrations and also batch corrections. And to our surprise, that looks very, very similar. We thought that's really fundamentally different, but they are not really strikingly different. But still, by focusing on that monocyte macrophage populations here, and we found there's a difference between healthy liver perfusate versus cirrhotic liver, and that increased some inflammatory Kuppa cell increase in the cirrhotic liver. So we are really digging into what kind of gene profile they have in the cirrhotic liver. And also, as I mentioned, we are doing a spatial gene transcript analysis by using NanoShrink cosmics. And so here we use several liver diseases and to analyze immune cells inside the liver tissues. So here I'm showing just one slide just to show that this is still ongoing effort for us. And we are able to identify the different clusters in the cosmics data with the different gene profiles. And we are kind of looking at neighborhood cells by using this data analysis. So maybe last 3 or 5 minutes or so, maybe I want to talk about the antigen-specific T-cell analysis by using liver perfusate. As I showed in the earlier slide, we found that basically liver perfusate contains many different types of immune cells, including antigen-presenting cells such as Kupffer cells and dendritic cells. And we thought maybe perfusate can be used to identify antigen-specific T-cell response, like the previous speaker showed, by using blood samples. So here in this study, so we asked whether the liver perfusate contain coronavirus-specific T-cells in the human liver. So here what we have done is basically we get liver perfusate from healthy volunteers, and we culture them with the spike proteins of the SARS-CoV-2 or human common coronaviruses, spike proteins. So as the previous speaker showed, by culturing the PBMC, or in this case liver perfusate, with the target proteins, so that activates the antigen-specific T cells. So this activated T cell can be captured by either flow cytometry or by sorting the T cells and doing single-cell sequencing. So we did that to understand what is the kind of feature of the antigen-specific T cell in the human liver. So here I'm showing the results. So here we are comparing the basically phenotype of the UMAP analysis of the coronavirus reactive CD4 T cell in the human liver as compared to total memory CD4 T cell in the human liver. I see that there's some difference shift, and we found that these populations, which is coded by the coronavirus-specific CD4 T cells, express high level of CD39, IGOS, and CCL6, which is really kind of unique feature. And also we wanted to ask whether that liver perfusate, that presence of that very particular kind of cells, are unique to the liver perfusate. So we compared the phenotype of the COV-reactive CD4 T cells between liver perfusate and the blood samples, and we found basically that the liver perfusate contains CD4 T cells express many high-level CD39 IGOS and CCL6 as compared to blood samples, as shown here. So here, I don't go into detail today, but these CD39 positive T cells basically found their Tregs in the space for the coronaviruses residing in the human liver. So what is the ongoing project? So because we collected many different liver perfusate samples from healthy volunteers, so we are asking the difference of the sex and the impact of aging in the liver resident immune cells. So because we have liver perfusate aged from 25 to 65, so we can analyze kind of aging effect and sex effect of the liver resident immune cells, such as monocyte Kupffer cells and also the T cells. So we are basically isolating these Kupffer cells and the T cells from each perfusate from all these 80 samples, and we're doing all single-cell data analysis. Asking, so what is in the liver monocyte Kupffer cells, asking what is the transcriptional shift by aging? Because the liver is constantly exposed to microbes from gut microenvironment. So as Dr. Okada showed in the previous slides, so basically gut microbiota is heavily affected by what we eat, right? So here we are, of course, the dietary changes and in the different ages. So we want to see the effect of the aging of the Kupffer cells, and also we want to see how much epidemic changes happens because of the aging in the Kupffer cells. And also Kupffer cells are derived from, it's known that they are from yolk sac derived cells or monocyte derived cells. So we want to know how much aging affects the origin of these Kupffer precursors shifting from yolk sac derived to the monocyte derived. So in the case of T cells, we want to know what is the emerging cell populations by aging. So again, so the T cell can be replenished in the liver tissue environment. So we want to know the effect of the aging. And we also want to know the clonal diversity, with the diversity of clonality going down, going up, or that's limited to some certain subsets of T cells. And also here, I don't talk much today, but we are really working on characterization of naive T cell in the human liver. So human liver is a very kind of unique organs as compared to other organs that contains many good frequency of naive T cells, Like in the case of CD4 T cells, approximately 10 to 15% of T cells are naive T cells, which is really surprising. And by doing analysis of naive T cells, we found that naive T cell in the liver somehow is different from a naive T cell in the blood or cold blood naive T cells. So we're really interested in the character of the naive T cells by aging, how much they are affected, what is the functional role for the protection of the liver from a microbial insult. So here, really, I'm interested in heterogeneity in this aging, but here I'm really heterogeneity, ethnicity, and geography. So basically, that's why I really wanted to join this HCA consortium and discuss about this with many audience, because geography and ethnicity is really key component to understand the biology of all the liver tissue region immune cells. So by doing this kind of same effort together, we can make a very good atlas of the human immune cell atlases. So conclusions, with a multiple platform single cell analysis approach, it is possible to efficiently and cost effectively analyze the immune system in a large number of samples. So establishing the impact of sex, aging, ethnicity, and geography in tissue-resident immune cells in healthy liver will provide significant insights into pathogenesis of the various liver diseases, including autoimmune chronic hepatitis, steatohepatitis, and cancer. So I'm going to stop here, and I want to acknowledge all my lab members, and including Yayoi and Hajime and Ma, Shuhei, and all of the lab members who contributed a lot for the sample collection, sample storage, and all these things. And all the samples in this talk is derived from hepatobiology and transpersonal department from Kyoto University. So I stop here. Happy to take any questions.
Professor Ueno, thank you so much for your great talk. So now it's open for discussions, questions. So go to the microphone and ask your questions.
Great. Thank you for your presentation. Can you explain more, what's the correlation between multicolor flow to single-cell transcriptome? If you were to compare the proportions of cell types I discover, what's the correlation between the two platforms? And also, because obviously I didn't see the neutrophils, which are key factors in single cell.
Right. So flow cytometry, as you mentioned, so neutrophils and the basophils, so this kind of granulocytes, it's really good platform to analyze in the liver tissues. But in a single cell, somehow, neutrophils is really hard to detect. So only we can detect in a neutrophil, in a single cell, is that the immature granulocytes. And we are kind of really testing whether that granulocytes reside in the liver is different from blood samples or not. Now we are analyzing that. So that's the one advantage, using combining flow cytometry together with a single cell.
But other than neutrophils, do you see good correlation in cell type proportions?
Yeah, thank you. So that's really, again, very, very fundamental insights. So we started doing this multiple single cell sequencing, but so far we are focused on the Kupffer cells and CD4 T cells. So we really didn't do side-by-side comparisons between flow and the single cell.
Thank you.
Yeah. Okay.
Fascinating talk. I just wondered how much of the heterogeneity of hepatocytes you're getting, because they're sensitive cells. It's frequently difficult to identify, but it's very interesting that you're able to identify quite a few. I saw hepatocytes, but I'm just wondering if you looked at the different subtypes.
Hepatocytes?
Hepatocytes, yeah, like portal, pericentral, periportal.
In the cosmics, you mean?
No, in the
single cell.
In the single cell.
Single cell. So basically, we are focused on CD4 positive T cells. So we're excluding hepatocytes.
Okay.
But somehow, yeah, that's fine. Yeah.
Okay. Thanks.
Hello. So I want to look at the tissue resident immune cells in the eye, and I was wondering why you didn't categorize them into CD69 and CD103 TRM cells.
Right.
Or did I miss the data? I don't know.
Yeah, so you have a good guy. So we are using CD69 as a kind of tissue, as a marker for the tissue resident memory cells.
Yeah.
But we start thinking that that's not only marker to define tissue resident T cell in the human liver. And we try to look for CD69 negative T cell compartment, and somehow we really start seeing that some other markers, or maybe involved for the tissue residency in the human liver. So yeah, maybe I can tell later.
Okay, thank you.
Okay, thank you again for great talk for Professor Ueno.
Thank you.
Okay, now let's move on to the last talk of this session. The speaker is Boxiang Liu, the leading analyst of the human cell across Asia and IU consortium. So he's going to talk single-cell sQTL link cell type specific splicing to complex diseases. Okay, Boxiang, please go ahead with your talk.
Okay. Thank you, Yuki, for the introduction, and I want to start by thanking the organizer for inviting me and also letting me infiltrating this infectious disease section, because this talk is not related to infectious disease, but it's more focused on single-cell sQTLs. So my name is Boxiang Liu. I am a PI at the National University of Singapore, and the topic today is single-cell sQTL links cell type specific splicing to complex diseases. It is well known by now, perhaps too well known, that the number of GWAS loci has expanded substantially between 2007, when the first comprehensive GWAS was done by the WCCC consortium, and now where tens of thousands of GWAS loci are known across the entire genome. Understanding the biological mechanism of GWAS is difficult because most GWAS loci are in the non-coding regions, and only 10% are in coding regions. Therefore, it's really important to leverage the tools of functional genomics to systematically dissect GWAS mechanisms. To understand the functional mechanism of GWAS loci, the GTEx consortium was set out to establish the first comprehensive QTL catalog across all the major human tissues, and the latest GTEx project sampled more than 800 post-mortem donors across 49 tissues. Because of the large sample size, GTEx has substantial power to detect eQTLs. So the latest version of GTEx shows that more than 97% of the SNPs are nominally associated with the expression of at least one gene, and that more than 60% of SNPs are associated with gene expression after accounting for the number of tissues. Even with this substantial power to detect eQTLs, one would expect that, perhaps close to 100% of the GWAS loci would colocalize with at least one GTEx eQTLs, but that's not the case. As shown here, less than half of the GWAS loci actually colocalize with GTEx eQTLs. So this naturally begs the question, where are the disease-associated QTLs, right? So in recent years, much of the field have reached the conclusion that to find disease eQTLs, we have to look at the right cell types, the right differentiation stages, and the right perturbations. And to magnify all these, we have to sample diverse ancestries, ethnicities, and also comprehensive molecular phenotypes. So, guided by these principles, significant efforts has gone into creating single-cell QTL maps. So this slide shows a subset of single-cell QTL papers, but it is easy to notice that all of these papers are focused on European populations, and all of them are focused on expression QTLs. This raises two questions. The first one is, what about non-European populations? As the previous speakers, like for example Sham and Parthas pointed out, the needle has not moved in the past 15 years, right? So most of the functional genomics as well as GWAS are focused on still European populations. And the second question is, what about other phenotypes? Especially, for example, splicing, because you can get that for free if you are studying expression already. So to answer these two challenges, we leveraged the Asian Immune Diversity Atlas Phase 1 dataset, which you have heard from Ken Hong before yesterday, which contained about 500 donors from diverse Asian ancestries from Japan, Korea, and Singapore. So Ken Hong from Sham's group identified 34 PBMC clusters, and 21 of which had a sufficient number of cells for downstream analysis. This includes major cell types, for example, B cells, T cells, natural killers, monocytes, and dendritic cells. So using a combination of experimental and computational pipelines, we're able to extract a median of more than 1,000 alternatively spliced genes per cell. As a benchmark, we applied the same computational pipeline to 1K1K, which was published last year on Science, and our detection rate is 4 times as much as 1K1K. So as you can see, I'm trying the pointer here. So this is us, and this is 1K. So to demonstrate that the increased detection rate of alternative splicing is not merely due to higher sequencing depth, we plotted the number of alternatively spliced genes versus the number of expressed genes, which is a proxy of sequencing depth. And we observed a higher detection rate for alternatively spliced genes across the entire spectrum, regardless of the sequencing depth. Now that we can capture a comprehensive set of alternatively spliced genes, we compare splicing differences across the three major populations: East Asian, Southeast Asian, and South Asians. So we identified hundreds of differential splicing events across 21 cell types. We found that ancestry bias splicing can be attributed to allele frequency differences. So here I'm showing one example. This variant disrupts the canonical splice site of exon 2 of the gene SBSB2. So I'm going to point out this to you. So this is the variant, and this is the splice site. So it changes this G variant to an A variant, thus eradicating the splice site. So this action forces the splicing mechanism to use a downstream alternative splice site 12BP downstream. So what's more interesting is that this variant has an allele frequency spectrum from East Asians to Southeast Asians to South Asians. And you can see that there is a corresponding reverse gradient from the East Asians to Southeast Asians to South Asians. And next, we can, because we have a large sample size, we can use pseudobulk splicing quantification to identify cis sQTLs as well as trans sQTLs. So here, by mapping QTL cell type by cell type, we are able to identify more than 11,000 cis eQTLs as well as more than 600 trans sQTLs across the 19 cell types. So I'm not going to go through every one of these QTL examples with you, but I think this example is quite interesting. So we hypothesized that some of the trans splicing effects can be mediated by cis effects on RNA binding proteins. And here I'll show you one example of how a cis eQTL effect on a master splicing regulator can have a distal effect on the alternative splicing of CD45, also known as PTPRC. This is a key gene that is involved in T cell development. So in the first step of this analysis, we identified a strong cis eQTL effect on HNRMP-LL in T cells. Interestingly, this cis eQTL effect on HNRMP-LL is a cell type specific one, with two distinct lead variants across the naive and memory T cells. Next, we performed the colocalization analysis and found a very strong colocalization between HNRMP expression and CD45 splicing across almost all T cell subtypes. So on this figure on the right, the bright colors means that we have found a cis eQTL effect on HNRMP-LL, and you can see correspondingly for these trans splicing QTL effect on CD45. And this shows the colocalization between the cis eQTLs and the trans sQTLs. As a orthogonal validation, we used single cell level expression and splicing quantification, and we observed a significant difference in CD45 splicing between cells that express HNRMP and those that don't. So these are the cells that are expressing HNRMP, and not expressing HNRMP, and these are the cells that are. And you can see that the isoform differences across these two groups. Finally, using Mendelian randomization, we observed that changes in CD45 splicing actually leads to phenotypic changes in the cell type proportions as well. So we can see that higher CD45 splicing actually promotes the activation from naive to activated T cell proportions. And using Mendelian randomization, we can see that the effect is highly significant. So before I go away, I just want to highlight, all these 3 steps of analysis uses a single data set, which really shows the power of large-scale single-cell data sets. So moving on from biological discovery to disease mechanisms, we performed colocalization between splicing QTLs and 20 GWAS traits, including 5 immune-related diseases. So here I'm going to highlight one putative target gene for Graves' disease. So this gene is called TCHP, and the colocalization between TCHP and Graves' disease GWAS is cell type specific. So the first panel shows Graves' disease GWAS, and this is the lead variant. And then this is the monocyte and NK cells. So the effect gradually decreases, and when you reach B cells, the effect disappears. So what I want to highlight here is that if you are not doing single cell and you're doing bulk sequencing, then you might not be able to see this because this effect is highly cell type specific. So not only is that a cell type specific effect, what I also want to further highlight is that this effect is an ancestry specific one as well. But before I go into that, I want to talk to you about where this variant locates. So this is exon 4 of TCHP, and this variant is located at the 5' splice site of exon 4, and it disrupts this splicing motif. And this variant is actually East Asian specific. So what I'm showing you here, so these guys are the Japanese population, Chinese population, and Korean population. So as you can see, the average allele frequency across the East Asian populations is around 10%, whereas if we go into the Southeast Asian populations, like Malay, this allele frequency decreases to about 4%. And if we move to South Asian populations, then the minor allele frequency drops to 0. And we can replicate this finding using 1,000 genomes. So here you can see that East Asians has about 10% of allele frequency, but in Europeans, Native Americans, Africans, and South Asians, there's no such alternative allele. Finally, we validated this effect using a mini gene construct. So here, what we did was we cloned out the exon 4 of TCHP, and it's flanking 200 BP. And the only differences between the 2 mini gene construct is this base. So as you can see, when we have the reference allele, then this returns a normal isoform. But if we have the alternative allele, then that results in intron retention and a heavier band. So I just want to quickly summarize of our findings here. So the first one is that ancestry biased alternatively splicing is widespread across the entire genome, and sometimes it can be explained by allele frequency differences, which are simple and easy to accept. And trans-splicing effects can be mediated by cis-eQTL effect on RNA binding proteins. And here I showed you an example between HNRP-LL and CD45, which is a key gene in T-cell development. And last but not least, TCHP, we think, could be a putative causal gene for Graves' disease. And what's more interesting is that its risk allele only appears in East Asian populations. And of course, this right now is a bit speculative, and more wet lab experiment will be needed. And I just want to thank all of my collaborators, without whom this project won't be possible. Especially I want to highlight a few people. So Tian Shi is my first PhD student, and he's responsible for processing the data set and alternative splicing analysis. And Yun Tian, who is right now giving a talk in South Africa right now, because I cannot be in 2 places at the same time. And he's responsible for the splicing QTL calling. And then Yi Han is also a first-year PhD student, and she worked on the dynamic QTL, which unfortunately I didn't have time to show today. And also our wonderful collaborators, Ken Hong, who generated the PBM cell type annotations, and the leaders of IDA who have generously provided us with the data set. And I just want to plug in one slide. So we're a fairly new lab. We just started last year, and we're recruiting at all levels, but especially we're interested in people who are well versed in AI and machine learning and who are interested in splicing. So here's my email and my website. If you're interested, feel free to get in touch with me after this talk or through email. So thank you very much, and I'll take any questions.
Lucian, thank you so much for your great talk. We're really impressed how you extract nice information in a way people didn't do it. So it's now open for discussion and question. People who have any? Yes, please go ahead.
That's really, really nice work. Thank you for sharing that. I was wondering how you actually define the ancestry-specific QTLs. Do you do something similar to the stuff that was done by Stephen Montgomery, where you use the regression approach, or do you take a different approach to defining ancestry specificity?
Yeah. So here we're showing one of the examples of the ancestry QTLs, but you're asking in general how we can define ancestry-specific QTLs. Yeah. So that's a great question. So one thing that we could do is just very loosely to use a different threshold for example, GTEx QTLs. So let's say our discovery is like 5%, and then we use a 10% cutoff for GTEx QTLs. And if it doesn't pass that threshold in GTEx, then we can call it specific for Asians. But I think that's a simplistic way that we have been using, but there are more advanced ways which I can talk about later individually.
I think the most important thing about your analysis to be able to leverage the 5' strategy to call these exon patterns. Could you maybe perhaps enlighten us a bit about how you were able to extrapolate these exon splicing patterns using single cell 5' methods?
Yeah.
How did you do that?
Absolutely. Thank you for that very nice question. And thanks to you, actually. So the reason we can do this is because of the 5' RNA data set. And we just want to give a shout out to the Riken people and also the IDA team for generating this 5' data set. Because when you use 5', there's something called exon painting effect. So basically that's a combination of strand invasion and also 5' stochastic breakage and recapping. So that means you can sample deeply into the exons without ever having to use the full length, right? So I don't have a slide here, but you can see we'll put the paper on archive soon that we can actually get to about 80% coverage by using the 5' data, which is really amazing. So that's how we actually leveraged the 5' data to get more coverage and to able to see deeply into the exon space. Yeah, thank you for that question.
Boxiang, very nice talk. Do you have any guidance for us in terms of an unknown disease, and if you're going to try out which particular cell type we should test it on? How do we know which cell type to choose? And secondly, if we are going to try out on all possible cell types, we'll run into multiple testing problems and we won't get anything to be significant. So do you have any guidance for that?
Yeah, I think that's a great question. So definitely if we have more cell types, then we run into multiple hypothesis testing burden. So here we're quite, I guess in a sense lucky because we only have blood cell types. But I guess you're asking if we had more tissues. So for example, the SC GTEx project that Sham and Christine is organizing, that would span obviously more than one cell type, more than one tissue. More money, I guess, more sample size. Perhaps, okay. So I guess if I were to do this, perhaps I'll choose the relevant cell type first. For example, using stratified LD score regression, and then focus on those cell types for the disease that you're interested. So basically, for example, as Jay showed before, you can do one sample first for tissues that you're interested, and then run LDS SLDSC for the cell type specific expressions, so to choose the cell type that you're interested, and then perhaps expand on those cell types using large sample sizes using a QTL study. Perhaps I'll do this in 2 steps. Yeah, but we can discuss later about this.
Thank you.
Okay, thank you again, Boxiang, for your wonderful talk. So so we wrap up the session, and thank you everyone everyone joining the session, and everyone speaker for being nice talk. Thank you for listening. Thank you.